Hey guys, and welcome back to Now I Know. Today we are talking about Enzyme Linked Immunosorbent Assay, which is also known as ELISA. Now ELISA is a type of immunoassay, and if you guys remember, we have talked about type of immunoassay RIA a couple of videos back. So it's an immunoassay, that means it utilizes, or the assay is based on antigen antibody interaction to detect presence of either antigen or antibody. Simple enough. And in RIA we saw that we actually detected presence of antigen or antibody by radioisotope, right? In case of ELISA, instead of radioisotope, we use enzyme for the detection, and thus it is named so, enzyme linked. Now since we are using enzyme for the detection, it's going to be less expensive compared to radioisotope, using radioisotope, and also it's going to be more safe. Now as I said, antigen or antibody detection, that means it is going to detect the presence of antigen or antibody. So it is qualitative analysis. Also we can find out how much, means it measures how much of that antigen or antibody is present. So it is also a quantitative type of assay. ELISA was given in 1971 by Peter Perlman and Eva Engwall. So now ELISA is also done in 96 well plate. Alright, and these are special ELISA plate, okay, not normal tissue culture 96 well plate. These are specially designed for ELISA. So 96 wells will be there, and if at all that antigen or antibody, whatever we are looking for, is present in the sample, this enzyme would help us in color detection, in color development. So if at all we observe color development, we can say that particular antigen or antibody is present in the sample. That's how enzyme is going to help us by color development. Detail we will see as we go on and see the mechanism. Now when we talk about ELISA, basically there are three variants that were developed initially, and we are going to talk about those three variants. Based on this, now there are a lot other variants that are, that you might read online. So basic principle would be the same, and I'm going to talk about three basic variants that we have in ELISA. So let's start with the first one. The first method is called Indirect Eliza, and this is used for detection of antibody in the sample. Sample like, say for example, serum sample. Now indirect ELISA is a method of choice, especially for, it is used to detect the HIV infection. Alright, so that means if a person has HIV infection, that person will produce antibodies against it. So if we detect this antibody in the serum, we can diagnose whether the person is infected with HIV or not. So what happens is, let's take it step by step. Now ELISA, as I said, we perform it in 96 well plate. So whatever I'm going to show is a picture of one well, okay, magnified view of one well. So what happens is, say for example, we are looking for antibodies for HIV, okay, antibodies against HIV. Now we know that we are looking for antibodies against HIV. So what we need to do is, in lab, we need a protein or antigen similar to HIV. Virus, maybe envelope protein or some core protein, so that this antibody, if at all is present, would go and bind it. So my first step would be to have recombinant HIV protein. Maybe let's say for example I might take core protein, or maybe envelope protein, anything. So I will immobilize that in my well, okay. I'm going to coat my well with that antigen. Again, why am I doing this? Because if at all the antibody is there in the sample, I need to detect it. So I need that antigen with what this antibody can bind. Now let's go ahead and add the serum sample of a patient in this particular well. We'll add the patient's serum, and if at all the serum has the antibody that we are looking for, it will go and bind to this antigen, right? This will be the first thing. My next concern is, how can I detect this? Whether it is present or not, how am I going to see that? So now every step that we are going to do, there will be a wash step in between. Okay, that looked like inverted heart. That's because we want to remove any unbound material in between the steps. So there will be a wash step every time that we add something. So now next would be for the detection, we are going to add something called a secondary antibody, and these antibodies are conjugated with enzyme. Okay, this is where enzyme comes into picture. And these secondary antibodies, what is the speciality of these secondary antibodies are they can bind to this primary antibody FC region. Okay, these are the anti-antibodies. Alright, so they can bind to the FC portion of this primary antibody, and they are labeled with enzyme, they are conjugated with enzyme. So let's first see that in the FC portion these secondary antibodies would bind, which is conjugated with enzyme. Okay, generally you can produce these secondary antibodies by from rabbit or sheep. Again, we have talked about it in radioimmunoassay. So this will recognize the human antibodies and bind against it. Okay, FC portion it can bind to. So when we add secondary antibody, they are conjugated with enzyme. So if there is an enzyme, definitely it will have a substrate on which it can work on. So here I am going to add colorless substrate. So if I add substrate over here, this enzyme has the ability to convert it into a color product. Okay, it will produce color. Okay, let me take a bright color. It will produce a color product. Alright, so in the end, what essentially I will observe in my plate, I will see color development if that particular antibody is present in the sample of a patient. Right, what we did, we are looking for specific antibody, so I produced recombinant protein for it in the lab. I have coated my ELISA well with that antigen, added my serum sample, antibody was there, it bound to the antigen. How to detect? I went on and added the secondary antibody, which is conjugated with enzyme, which has the ability to convert the colorless substrate into color product so that I can visually see it, I can visualize it. So later on I added the colorless substrate. Because antibody was present, secondary antibody bound to it and it produced the colored product. So at the end, I observed the color development, and thus it gave me positive result. The person is having HIV infection. Right? Now, this was about the detection. It just gave me simply positive or negative result. I can also measure the quantity, because the intensity, generally what happens when we take a sample, we just don't take directly the sample and add. We generally go on and dilute the sample, and then we perform this test for that diluted number of samples. So in the end, I might observe a different variation. The intensity of color will be different depending on how much of antibody is present after dilution. So that I can quantify how much of my antibody or even antigen for that matter is present. But that part I will come to it in the end of a video. Right now let's just understand the principle, what is happening in every type of variant. So this is how I can find out the detection of antibody. Now here we are adding primary antibody, that is a sample, and then we have the secondary antibody, and that binds and gives us the result. In that sense it is called indirect ELISA. So now let's move on to the second variant, and we'll see how antigen presence can be detected. So Sandwich ELISA is for detection of antigen present in the sample. You can detect any protein, peptides, hormone presence in the sample. So now it should be easy enough to understand. If you're looking for antigen in my lab setup, in my ELISA plate, I would want antibody against that, right? So let's go ahead and make that. I would have my well of plate, sorry for the diagrams, coated with the antibody against the antigen that I'm looking. We'll go ahead and add the sample. So let's add the patient sample. If the sample is having that particular antigen, what will happen? It will go and bind to this antibodies, right? Again, how to detect it? You guessed it right, we are going to add secondary antibodies. which are conjugated with enzyme. So if at all the antigen is present, this secondary antibody will go and bind with it. And detection is possible by adding colorless substrate. Enzyme will give us colored product in the end. Alright. Now because here the antigen is sandwich between two antibodies, it is called Sandwich ELISA. Whatever antigen that you're looking for, coat your ELISA well with that particular antibodies, antibodies against that antigen. Add the sample. If at all it has the antigen, it will bind to it. Add the secondary antibody conjugated with enzyme against it. So if the antigen was there, it will go and bind to it. Add the substrate, colorless substrate. In the end, you will observe color development in the plate. So that's how Sandwich ELISA is done. Now let's move on to the third type of variant. The third variant is Competitive ELISA, and that is also for detection of antigen. Now you might be wondering, we already seen one method for detection of antigen. Why do we need one more method for that? So that I'll tell you once we are done. I'll tell you why do we need one more variation. So what happens in Competitive ELISA is, I know I'm working with particular antigen, right? So I need antibody against that. So I'm going to have particular antibodies against that antigen, and then I'm going to mix the sample with this antibody. Now this step is not done in ELISA plate, okay? This is done before we add this mix to ELISA plate. So this is outside the ELISA plate, okay? And these antibodies are against whatever antigen that we are looking for. Okay, so if at all antigen is present in the sample, it will go and bind to these antibodies. Say for example only two antigens are present, it will go and bind to one antibody, because there will be two antigen binding site present on one antibody. So it will go and bind to this one antibody, and this mix now we are going to put it in the ELISA plate. Now here in my ELISA well, instead, here for detection we had immobilized antibodies in the well, right? Here instead of that, I'm going to have the same recombinant protein, the same antigen that I'm looking for. That same recombinant antigen I'm going to immobilize in my well. You just go with it. You will understand why am I doing this. So what I did first, took my sample, mixed it with the antibody. This antibody is against whatever antigen that I'm trying to detect. If the antigen is present, it will go and bind to this antibody. Simple enough. Here in this case, I have one antibody extra left. So now I have this reaction mix. Now I will add this reaction mix in the ELISA plate. And here the wells are coated with the recombinant antigen that I'm looking for. And what will happen when I add this mix into it? Look at this. This antibody is already bound with my sample antigen. So if I add this over here, this antibody does not have any free antigen binding site to bind with this well, right? So this antibody will not bind because it is already bound by my sample. But there is one more antibody that has free antigen binding site. So that will go and bind with this recombinant antigen. Now I'll go and wash this well. Now I will go ahead and wash these wells. What will happen? This bound antibody will get washed away and only one antibody is bound over here. Now I will go ahead and add my secondary antibody because I need to detect this. Now you must be wondering, the one which was bound with the sample is already washed away. Why am I doing this? That also we'll understand just in a while. Let's go ahead and add our secondary antibody conjugated with enzyme. Add the colorless substrate. It will produce the colored product. Now what will happen is, the intensity of the color that we are going to observe in this case would be actually inversely proportional to the concentration of antigen in the sample. Let me slow down. What I'm trying to say over here is, say for example the sample had higher amount of antigen. there was more amount of antigen, what will happen? More amount of these antibodies will be bound by my sample antigen, and less antibodies would be available to bind to the recombinant antigen present in the well. So as a result, less amount of secondary antibody will bind and less amount of color produced. The intensity of the color will be less. So higher the concentration of antigen in the sample, lesser the intensity of a color in the end. Right? I hope I'm not confusing. If there is low amount of antigen in the sample, means there are many free antibodies to bind to my well. Many secondary antibodies would bind, there will be more of these primary antibodies, so color production will be more. So in case of antigen concentration is less, there will be high intensity in the end. So that's what happens in competitive ELISA. Higher the concentration of antigen in the sample, lower the intensity in the end, and if the antigen is less in the sample, there will be more intensity in the end. Now you must be wondering why to go for such a complicated method when we already saw Sandwich ELISA. Now you see this Sandwich ELISA, at a time to one antigen there is two antibodies binding. That means on that antigen there has to be at least two epitopes. Once again, epitopes, if you guys remember, these are the antibody binding site on antigen. So there has to be two epitopes at least to which the antibody can bind, two antibodies can bind. Also the antigen should be big enough to carry those two epitopes. But not all antigens are that big enough, or not all antigens would have two epitopes or more epitopes. Now, what am I talking about? Once again, we have talked about something known as heptanes, smaller antigens which are so small that they will not have more than one epitopes for two antibodies to bind. So how can I detect it? For such smaller antigens, this method is useful. Competitive ELISA is useful because there is no way that two antibodies can bind it. So when it comes to antibody, we can go ahead and do the indirect ELISA. And when it comes to antigen, depending on what type of antigen we are dealing with, you either go for Sandwich ELISA or competitive ELISA. Now one more question comes in my mind when we talk about competitive ELISA is we are using here antibodies in lab, primary antibody which is not conjugated with enzyme, and then we are adding secondary antibody which is conjugated with enzyme for the detection. Can't I directly label this primary antibody itself and add the substrate in this step? Why do I have to add primary and then go ahead and add secondary antibody? Like it's one more extra step. Why do I need to do that? So the basic, like, you can do it, okay? It's not like we can't do it, and it's done also. Direct ELISA, that is competitive direct ELISA, direct competitive ELISA. This is indirect type. So why this indirect type is preferred is the first and very important advantage of using this secondary antibody conjugated with enzyme is when we talk about the secondary antibodies, these antibodies are, they can bind to any type of primary antibodies. Okay, large number of primary antibodies, it can bind to Fc region of any type of primary antibodies. Okay, so with one set of secondary antibodies that I have conjugated with enzyme can bind with any type of primary antibodies. And when I say any type is because, see, I'm looking for antigen in the sample. This antigen would differ every time based on what I'm looking for. Every time I'll have different type of antigen to be detected. That means every time I need that particular antibody also against it. So I need to have specific antibody, primary antibody that can bind with it. So every time my primary antibody will be different. Now imagine this itself is one big task to produce antibody against it. If I have to label every time, if I have to produce conjugated primary antibodies, is going to increase my work, is going to increase the cost of that production. Every time I'll have to have specific conjugated antibodies for different antigen. Instead of that, I can have secondary antibodies which has the ability to bind to Fc portion of antibodies, because these are, as I said, let's say for example they are produced in rabbit, they can bind to human antibody assay region, to any human antibody, right? So if I have one set of secondary antibodies conjugated with enzyme with different type of assay that I'm performing for detection of different type of antigen, I can go ahead and do multiple detection with this secondary antibody itself, right? So that becomes much more cost effective to design the assay. So that's why we use secondary antibodies. It is not always that you have to use it. You can always go ahead and label your primary antibody also. That also is, you might read about this competitive direct ELISA where directly primary antibody is labeled. Second important advantage over here would be, if you look at this, there will be two FC portion, right? Two side of this FC portion where the secondary antibody can bind. So to one primary antibody, two secondary antibody can bind. So the intensity of a color will be much more better. That means what I'm trying to say is it will increase the sensitivity of my assay. The color development will be very much better. So it will increase the sensitivity of my assay. So these two reasons are there why we go for indirect ELISA, okay, why we go for secondary antibody labeled with or conjugated with enzyme. Now that's all about how to do the detection. Now how can I quantify how much of this antigen or antibody is present in the sample? How to do the quantification of how much of antigen or antibody is present? So this also we have talked in detail when we were talking about radioimmunoassay. We saw that we need to produce a standard graph for that, okay? So I'm not going in that much detail over here. If you guys want, I'll put the link on the screen, just have a look. But essentially what we are doing is we are going to produce a standard graph. So what happens when you produce a standard graph? Once the assay is done, once we have the plate ready with the color development, we take this plate in specific ELISA readers, which will, based on the intensity of a color absorbance, we will get particular optical density. So we'll get particular OD at the end for every single well. So what happens is we produce a standard graph. If I'm working with some antibody, produce specific standard graph for that antibody. Working with antigen, produce a standard graph for particular antigen. So I'll have a standard graph. And as I said, for sample, dilute the sample, because if at all there is a very high amount of antigen or antibody, it should fall under this standard graph. So dilute your sample and check for the optical density versus concentration. So your standard graph would have specific OD based on the concentration of that standard. Now go ahead and take the OD of your sample. It will give you some OD. Check for the concentration. So some amount it will give, right? So this is the concentration of my sample. Since you are diluting, do not forget to multiply with your dilution factor after you have got the concentration. So that's how you can quantify in the end what is the amount based on the standard graph preparation. It's very simple to understand. So that's all for now. That's all about ELISA. You might read different variation, as I said in the beginning. Principle will be essentially the same. So that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every week, and I'll see you next time. Until then, keep learning.